الله الرحمن الرحيم رب اشرح لي صدري ويسر لي امري واحلل عقدة من لساني يفقهوا قولي. So first of all, I'm really grateful to the organizers for inviting me and giving the opportunity to share the state-of-the-art work which involves the biophysics, physics, biochemistry, molecular biology, immunology, as well as, of course, the field to which basically I have been field by Professor Naim Khwaja sitting here. So I admit here before start my talk, I'm not a veterinary scientist and neither have too much experience in immunology or the vaccinology. And actually, I turned to this field by chance. I'm a biochemist and a structural biologist, study the protein function and the mechanisms and how the protein function in living cells. So while studying such kind of the mechanisms, I turned towards the poultry viral protein as well as the animal pathogenic bacterial proteins. And then I found this is a very fascinating field where there is a lot of work need to be done with the advanced technology. And by chance, as penicillin was discovered, we discovered the few domains of the pentone-based protein of fall adenovirus that turn into a very, very effective vaccine. And with the help of Professor Naim Khwaja, and we have made large trials in Pakistan, and those are very, very successful. So by getting into the present, how much time I will have? So, so I will just give you the glimpse how the different scientific disciplines actually turn into the development of the applied products. So for the young students, I would say when you do a PhD particularly, it does not mean that you have been trained in a specific subject. Learning goes on whole life. And this is the case with me. I have been trained in the UK while doing PhD and the postdoc at Harvard Medical School to study the structure of the membrane proteins. But those techniques I applied towards the applied ventures in the poultry, pathogens. And now we are, I would say, in Pakistan, Alhamdulillah, have the mega grants from the World Bank of the Pakistan for the development of the recombinant as well as the subunit vaccine. So looking into this slide, actually, we work basically on membrane proteins. And today my topic, because there are many people involved, you will see in the last slide, to work on this project. Due to the shortage of time, I'm not getting it to introduce all of these. Only I will focus on the fall adenovirus and the infectious bursal disease virus. And this is the slide which I show always because I'm a biochemist. We take a gene and clone into multiple vectors and then express the proteins in E. coli, yeast, and very much in the cell lines. And then we get the purified protein and characterize them by structural means as well as by functional means. So when we discovered one of the important vaccine for the fall adenovirus 4 subunit vaccine, then we became interested as well in the, research which involves the generation and the production of the viral vector vaccines. And how this research is being carried out, we purify the recombinant proteins and crystallize them. And here you see the worldwide sources of the synchrotron. Synchrotron basically is a big machine which produces the radiation, and those radiation hit the crystals of the protein, and one can get the information about the structure of a molecule at the atomic level. So by having this lot of support worldwide, we get a lot of funding for the training of the students as well as to study the protein. And these are some players in structural biology to whom we collaborate actively, and we do the preliminary research in Pakistan, and most of the students then go abroad on the foreign funding in the laboratories of these, well-established structure biologists, and then we study the structures of the protein with their applied ventures in drug discovery, in vaccinology or the immunology. And the poultry, you are very well aware. I'm not getting into the details. What is its importance? This is a slide for the importance of poultry in Pakistan, and already addressed very well. And coming to this, you see the hydropericardium syndrome. When it occurred, mortality rate is very high, 80 to 100%. And likewise, IBDV, it is 60 to 90%. And this disease actually, HPS, all you know, first time reported in Pakistan, and the causative agent is fall adenovirus 4. So in addition to the fall adenovirus 4, in Pakistan, several laboratories, including the Aviros laboratory, they have reported the occurrence of the fall adenovirus 2, 11, and the fall adenovirus 8. So in today's presentation, I will only cover the fall adenovirus 4. But we have all the subunit vaccines available at the laboratory scale for the fall adenovirus 2, 11, and the 8 as well. And this slide, I'm just showing you the symptoms when hydropericardium syndrome occur. So most important symptom is the pericardial cavity around the heart is filled with some kind of the solution. So this I will show you at the end in my vaccine trials. You will see our vaccines prevent such kind of the disease symptoms. So this is the overall structural view of the fall adenovirus 4, where you see the three structural proteins, fiber, hexon, and the penton base. We were very much interested in the fiber and the hexon, but these don't express very well in the bacterial system. And also some of the structure were available. So we started this project to solve the structure of the penton-based protein of the fall adenovirus, and we ended up with the development of the subunit vaccine. And here you see the further elaboration on the penton base. It is a pentameric protein. This structure is actually constructed on the basis of the homologous structures from the human adenovirus. And this protein, if you further look into the deep at the secondary structure level, so it has an insertion domain, which is exposed on the outer side of the virus, and the jelly roll domain, which is on the inner side of the virus. And this is the whole full-length penton-based monomer. And because we were very interested to crystallize this protein, we express it, purify it. We were not getting success in order to get the crystal. So we then started work to produce its various truncated or the different length of the proteins in order to find the stable core of this protein. So for this, we use the bioinformatics program and to predict the structure of the protein as well as the sequence. And here you see in the red color, the amino acid 2 to 5, we find these amino acids are having more T and the B cell immunity-based epitopes. And we quickly express in E. coli. And here you see we get a lot of the protein. And then we went on to the immunization trial. But this part of the protein 1 to 25, which we may call it subunit vaccine, give only 50% protection against the fall adenovirus 4 challenge. So then we further study the ELISA and the other thing that we did publish this work in virus research. And in order to find the more potent and more stable domain of the penton base, so then we extended our studies with the help of mass spectrometry-based methods. So what we did in this, we took the purified protein, penton base, and we digested with the protease. We chopped the protein at the different arginine or lysine position, which are exposed on the surface of the protein. So by doing this, what we are doing, we are actually basically cleaving the loop region of the protein, which are not stable, and was looking forward for the stable core part of the protein. So we did this experiment at different time intervals and different enzyme concentration of the trypsin. And then by using the mass spectrometry methods and as well as the computation methods, we found this red part of the protein is a stable core of the protein. So we reclone it into the E. coli expression vector. And in addition to that, we also predicted the other domain of the protein, which are listed here, from asparagine asparagine 77 to glutamine 528, and the cysteine 143 to proline 474, and the cysteine 143 to the glycine 384. So at that time, our purpose was to find a stable core of the protein for the three-dimensional structure of this protein. Unfortunately, so far, we are not able to solve this three-dimensional structure of the protein, but we got a lot of success to use these proteins as a subunit vaccine. And here you see in all from fowlerinovirus 1 to 11, this insertion domain is very much conserved. So it means this can be used, this part of the protein as a subunit vaccine or for the characterization of to study the function of the pentone base among the all serotypes of the fowlerinovirus 4, fowlerinovirus. And this is the structural preview that it is a conserved. And then we expressed all the truncated version in E. coli. And here you see one thing which is very important, because if you want to use some kind of subunit vaccine as a vaccine in industry, you must be able to produce in large amounts. So when we started with the full length protein, you see, we were only getting 1 milligram per liter culture volume. So when we made different stable variants of these proteins, so we reached up to the 42 milligram. And now we have devised the method in which we produce up to 500 milligram per liter culture volume. And we have also simplified the purification techniques that allow us to apply these proteins, at large, vaccination level. And these are the structural techniques which we use to the stability of the protein. And because we do structure biology, so the one of the stable domain is a pentameric. It means it retains the physiological condition, which is one of the prerequisite to be an effective vaccine for any molecule. And because this protein was not crystallized, we take the advantage of electron microscopy, electron crystallography, and we study the single particle. And here you see the pentameric nature of the protein. And this work is still going on because we are not finding the homogeneous particle. And we did see some hexameric structure, which is not physiologically can be justified. So then we take all these three truncated version of the protein, and you see we get 100% protection against the fowlerinovirus pathogen. And then we also determine the antibody titer using the subunit vaccine as an antigen, and also the whole virus to verify, okay, this protein basically can neutralize the virus as well. And then the most, I would say, promising thing in this subunit vaccine is, you see, we store this protein in 50% glycerol for over a period of 1 year in freezer. And now we have the data to store this protein over a period of 3 years. And it's still immunogenic, it still works, and it is still as effective as a freshly purified, freshly generated subunit vaccine. So this is one of the, I think in case of the subunit vaccine is the advantage because you can keep it. And now we have its, formulation in the lyophilized form. That may be stable up to 5 years that we have not tested so far. And similarly, this is the other variant. And this all work we have established. I never ever hide my work. This is my first, decision of life that I have been done. I share all the sequences of the proteins at the all forums. I published my all work, and I'm not very fan of getting patent. Only for a few grants there was a prerequisite. So I'm a believer. Anyone who can do research must come up in the field, and then if someone is able to do more work than myself or my group, then it is more happier for me. And because such kind of the work is not easy and not easy to compete. So therefore, all the data is available online about these kind of the vaccines. And then side by side, when we get some success with this kind of the vaccine, so we work on the viral vector vaccine. So due to that reason, because these are not very, these for basic point of work point of view, virus-like particle vaccines. So they are very fascinating, but we can't produce it in large amounts for poultry and the other stuff. So that is the reason, one of the bottleneck, although we did this work, we published, we find some epitopes very, very immunogenic and the protective, but from applied point of view, it is not possible to me to commercialize such kind of project. So various kind of the virus, components are used to make the virus-like particles, and we actually use the hepatitis B core protein for this work. And we take the hexon protein of the fall adenovirus 4, which is one of the most immunogenic protein in case of the fall adenoviruses. So, and we selected a number of regions. You see here the red and the pink and the light sky color here, part of the hexon protein. And we clone them, and you see we made a number of the constructs and then produce this protein, express them in E. coli, and then we purify them, then we do the challenge trials. So one of the epitope region you see here from aspartate 348 to phenylalanine, it gives 90% protection. And we also did postmortem studies, and with the help of the Professor Kashif Salimi, we also did some histopathology. So all results prove that this is an effective epitope. So again, but it can't be produced at large scale. The beauty of this epitope is it is very well conserved among the adenoviruses and can be used for making the vaccine for the other serotypes and the other adenoviruses. So, kitna time again? Couple of minutes, quickly. So in addition to that, viral vector vaccine. So we use the same vector which was used for the vaccines, SARS-CoV-2. So this is a non-replicated adenovirus 4 using the hexon base and the fiber 2 because it can't be expressed in the bacterial system. We generated the viruses. We did some qPCR. We did TCID50 for the dose determination and did the experiment. And this slide shows the postmortem trial where you don't see the pericardial fluid in the pericardial sac around the heart. So only one chicken has the infection in compared to the control where all the chicken have the infection on the pathogen challenge. So this I will just give you in 2 minutes the overview of the IBD. So here we use basically subunit protein-based vaccine as well as the viral vector-based vaccine. And this is the lab trial which shows the viral vector vaccine for the VP2 are the effective for the control of the IBD virus. And then we did a lot of the experiments and also the virus neutralization test, which proved that it can neutralize the antibodies. And then we did these trials at the atom and pharma independently, and you see the titer, which is a protective more than 2000, and there is no adverse effect on the body weight of the chicken. And we are optimizing bioreactor condition to produce the vaccine at large scale, but this is just under trial at the Center of Excellence in Molecular Biology. And we made multiple variants of the VP2. You see, produce the protein in Pichia pastori yeast, full length in E. coli, and it's one of the domain in E. coli as well, DNA vaccine, vector vaccine, and did comparative analysis of all these. All of these, most of these give 100% to 90% protection, but again, the subunit vaccines are expensive and difficult to produce. And we went on with the, viral vector vaccine. And these are the comparative trials from the commercial sector as well as our in-house ELASA, which, predict that this is the effective vaccine against the IBD. And we did develop the lateral flow assay for the detection of the antibodies against the VP2 protein. And these are the major funding where we have exported in millions of the rupees from the World Bank and the government of the Pakistan. And these are the collaborators, many. I'm not getting into the details of all this. And my mentor, Professor Munir Iqbal, is also sitting here, and he is advising me since last 10 years on the different aspects. And now we are in some basic position where we can turn into the applied work. And these are so many funding agencies that contributed directly or indirectly to this part of the work. And these are the three institutions mainly now contributing at the NIPG School of Biological Sciences and the Center of Excellence Molecular Biology. Last but not least, without this, it was not possible to accomplish this work at the applied level. So I am really grateful to the Professor Naeem Khwaja, who always encouraged me on the new projects, particularly where even people don't believe in the West. He believes in Pakistan that this could be the future of the vaccine, of subunit vaccines in the future. Thank you very much.